As I said in a previous video, you do not need to have a lot of prior knowledge of chemistry and biology to follow this course, as I will present all the necessary basics whenever necessary. So in the next few slides, we are going to have a closer look at what proteins are. Proteins are large biomolecules responsible for a vast range of functions in all living organisms, including catalyzing metabolic reactions, replicating DNA, the genetic code of all cells, responding to stimuli, providing structure to cells and organisms, and transporting molecules from one place to another. Proteins are made up of long chains of amino acids. There are 20 different amino acids that can be present in such a chain, and each amino acid is a small molecule. On this slide, you can see an example of an amino acid, the amino acid alanine. You can see that the amino acid is made up of different atoms bonded together. Later in this course, we will have a more detailed look at what small molecules are and how the different atoms are linked together, but for now, you just need to know that amino acids are small molecules. So proteins are long chains of amino acids, and successive amino acids are linked together by chemical bonds that are called peptide bonds. Such protein chains typically consists of a few hundred amino acids. When a protein amino acid sequence is built, the protein sequence does not remain a linear sequence, but instead it folds into a 3D structure. This folding occurs because of the attraction and repulsion between different parts of the amino acids in the chain. Some of the amino acids like to be close to water, which is typically present in the environment, and some parts try to avoid water. Because of these preferences, the protein chain bends and twists so that the water-avoiding parts are more inside, away from the water, and the water-loving parts are exposed to the water on the surface of the protein. This process is also influenced by bonds and interactions between amino acids that don't have to be close or next to each other in the linear sequence. All of these forces together cause the protein to fold into a specific 3D shape. The shape is very important because it determines the function of the protein. So, depending on the amino acid sequence, the 3D structure of a protein is more or less defined, and so is the function of the protein. And even a single change in the amino acid sequence can cause a protein to fold differently, become unstable, or lose its function. We will now take a closer look at how the protein is folding into its 3D structure. Every protein starts as a long chain made up of individual amino acids bonded together. We call this the primary structure. These amino acids then interact with their neighbors, causing the chain to twist and fold locally into specific shapes, such as coils called alpha helices and flatter sections known as sheets. And we call these structures the secondary structure. And finally, all these local sections, the helices, sheets, and the loops connecting them, pack and fold together in a specific arrangement, creating the protein's overall complex three-dimensional shape, which determines its function. And we call this the tertiary structure. Now you might wonder, where does that initial chain of amino acids, which we just saw folding into a protein, actually come from? This illustration shows the process begins with the DNA in the cell's nucleus, which holds the blueprint or the instructions on how to build the amino acid sequence. First, during a process called transcription, the relevant section of DNA, which stores the instructions for thousands of different proteins, is copied into a messenger molecule, mRNA. So this mRNA now stores only the information for a single protein. This mRNA is first transported outside of the cell nucleus, and the mRNA message is then read by a cellular machine called ribosome in a process called translation. The ribosome moves along the mRNA, linking together amino acids in specific order, dictated or encoded by the mRNA, creating the chain that then folds into the functional protein that we have discussed. Finally, I'm showing you a visualization of the process of how the ribosome links together amino acids and how the sequence then folds then folds into its 3D structure from a YouTube channel called Sprong Studio. You can see that as soon as the first amino acids are connected, local secondary structures begin to form, and finally the protein folds into its tertiary 3D structure. As I said, if we have different protein amino acid sequences, these will fold into different protein 3D structures with different functions. In every cell, there are thousands of different proteins that all have different functions. For example, in a simple bacterium such as E. coli, which is an organism consisting of only one cell, there are approximately 3,000 different proteins present and a total number of approximately 10 million proteins in each cell. All these 3,000 different proteins have different structures and functions responsible for producing all the relevant molecules for cell growth, for taking up molecules from the environment, for producing new proteins, and for giving the cell its structure. In all of your human cells, for example, your skin, blood, or brain cells, there are even more different proteins present, approximately 10,000 in each cell. The image on this slide hopefully gives you an idea of how diverse the resulting 3D structures can be for different protein sequences. On the next slide, we will look at some different functions that proteins can have. One important and large class of proteins are enzymes. Enzymes are proteins that can catalyze chemical reactions, which means they can massively speed up chemical reactions up to over 1 million times or more. Most chemical reactions that take place inside biological cells could also take place without enzymes, but they are much less likely to happen and it would take far too long. This image shows an example of an enzyme, lactase. Lactase is an enzyme that catalyzes the splitting of lactose, a milk sugar, into smaller sugar molecules, glucose and galactose. This means that the enzyme lactase helps to break down the lactose molecule, a sugar found in milk and dairy products, into smaller molecules that can be further utilized by other enzymes. The lactase enzyme has a binding pocket in its 3D structure where lactose can bind to the enzyme. This helps to break down lactase into molecules that our bodies can easily use. For people who are lactose intolerant, this chemical reaction of breaking down lactose into smaller molecules does not happen fast enough because the lactase enzyme is not produced often enough, and hence the cells cannot use the milk sugar as an energy source. As a result, undigested lactose remains in the gastrointestinal tract, causing a variety of symptoms such as stomach pain and diarrhea. The small molecules that an enzyme can bind to and can convert are called substrates. There can be one or more substrates in every enzyme catalyzed reaction. The transformed molecules that are the result of the chemical reaction are called the products. Like the substrates, there can be either one or more products. In this case, we have two products, but it could also be the case that we have an enzyme that only changes the structure of a single substrate, resulting in a single product. It is important to note that the enzyme is not altered in the process. This means that the enzyme binds to the substrate, transforms the substrate into products, releases them, and the enzyme has not changed during this whole process, which means that it can bind to a substrate again and catalyze the same chemical reaction. In every cell, there are many different enzymes that catalyze different reactions. In humans, for example, there are at least 3,400 enzymes with different functions. On this slide, I've listed some of the major classes of proteins to give you a better picture of what proteins can do. We saw an example of an enzyme on the previous slide. Another example of proteins are transport proteins, which are proteins that can transport molecules across membranes. For example, the cell membrane separates the inside of the cell from the outside of the cell, and cells need to be able to take up molecules from the environment and also be able to transport molecules from the inside to the outside of the cell. This is accomplished by transport proteins. The next example is regulatory proteins. These proteins regulate the activity of genes and gene expression to ensure that the right proteins are made at the right time and in the right amounts. In this example, you see the tumor suppressor protein P53. When DNA damage is detected, this protein can stop cell division and activate other proteins involved in DNA repair. And the last Example of a class of proteins that I will show are structural proteins. These proteins provide support and shape to cells and organisms. One example is the protein collagen, which looks like long, strong cables or ropes. We find collagen in our skin, bones, and muscles. For example, it helps to give our skin strength and elasticity, which allows the skin to stretch without breaking and then returning to its original shape. Hopefully, now you have a rough idea about what proteins are and what their functions can be. So, how can we store information about the protein? We have seen that proteins are sequences of amino acids. We also know that the amino acid sequence of a protein determines its structure, which determines its function. Thus, in principle, the amino acid sequence contains all the information we need to know about the protein. So we can represent the protein by its amino acid sequence, which is the most common way to store protein information. A major advantage of storing protein information through its amino acid sequence is that the amino acid sequence is readily available for most proteins of interest. For example, the UniProt database stores over 250 million different protein sequences. The amino acid sequence of a protein is usually represented by a sequence of capital letters, where each letter encodes an amino acid. This is very compact and storage efficient, since you only need to store one string for each protein. Here you can see all 20 amino acids that can be present in a protein. In this example, we would have a protein with alanine in the first position, leucine in the second position, and so on. This amino acid sequence already contains most of the information about the protein structure and function, so this is the way we will usually represent proteins in this course. We can store protein amino acid sequences in FASTA files. The FASTA format is a text-based format, which means that you can open and edit it with any editor or any text editor. Each entry in a FASTA file starts with a greater than character, followed by a description of the protein in the same line. In this example, on this slide, you can see that it simply says sequence 1 and sequence 2, but in principle, we could add any information we want about the protein to this line. In the following lines, we have the amino acid sequence of the protein. Each line usually contains no more than 80 amino acids, but this is just a convention and can be different. All amino acids, until the next entry starts with a greater than sign, belong to the same protein. We will see later that we can use these protein sequences as the input for transformer networks to process the sequence and to make predictions about the protein. Or alternatively, in the next video, we will see that we can use more traditional methods to extract information, properties, and statistics from the protein sequence to numerically represent the protein. You probably remember that the linear amino acid sequence falls into a 3D structure. So you may wonder why we do not encode the protein 3D structure instead of the linear protein amino acid sequence. If the protein structure is known, we can do that. We can determine the protein 3D structure either experimentally, which can be difficult and time consuming, or by predicting it using deep learning models such as AlphaFold, RosettaFold, or ESMFold. We can store this 3D structure using PDB files. PDB files are text files that contain the 3D coordinates of all the atoms in the protein. In this example, you can see that valine is the first amino acid of the protein and the coordinates of the valine atoms are given. This is much more memory intensive, but also more accurate and stores much more information than simply using the amino acid sequence of the protein. However, currently most methods for numerically encoding information for large numbers of proteins are based on the protein's amino acid sequence, and so we will usually represent proteins using their sequence. But towards the end of this lecture, we will look into some promising approaches that actually encode the protein 3D structure. One example how this can be achieved is displayed here. You can encode the protein as a 3D object, and then you can use it as the input to a three-dimensional convolutional neural network. 3D neural networks or convolutional neural networks work in the same way as 2D convolutional neural networks work for image processing, with the only difference that we are now in a 3D space and not in a 2D space.